Hello AP Biology students, this is going to be the fourth section in Unit 3. And in this section, we're going to talk about something called inhibitors. Just like their name implies, enzyme inhibition is going to occur when molecules are going to bind to the enzymes and decrease their activity. Now, there are a couple different types of enzyme inhibition, but again, they're binding to enzymes in order for them to stop the enzymatic reaction, or at least slow it down. Our first type of inhibitor is called a competitive inhibitor. This is where the inhibitor is going to bind with the active site and actively compete with the substrate. You can see in our normal reaction, our enzyme is going to break this molecule apart. But in our inhibited reaction, this inhibitor is going to come in and bind with that active site and not allow the substrate access to the active site. It's important to remember that in this type of inhibitor, the enzyme is not destroyed. In this type of inhibition, the inhibitor is binding with the active site. Classic meme. Enzyme, is this a substrate? No, it's not, but it looks like one. Before we get to our second type of inhibitor, we're going to learn about something called the allosteric site. This is a region away from the active site that's going to influence the active site. So you can see, again, we have our active site. This is where the substrate goes. Our allosteric site is going to be away from the inhibitor and it's going to bind with some molecule. Non-competitive inhibition is when an inhibitor is going to bind to the site called the allosteric site. This reaction, again, the binding of an inhibitor with the allosteric site, is going to change the active site in some way, thus regulating the enzyme. You can see here we have our enzyme, our substrate, and inhibitor. Our inhibitor is going to bind to that allosteric site and change the enzyme's active site, thus changing the enzymatic reaction. Usually it stops it. We also have something called allosteric activation, where a molecule is going to bind to an enzyme and activate it. This means that the enzyme is usually shut off unless an allosteric activator is going to bind to the enzyme. This is going to in turn regulate how much enzymes are on or off to help the efficiency of a cell. In the allosteric activation types, the enzyme's active site will change once that activator bonds to the allosteric site. Again, high quality meme, 10 out of 10.